Okay. And one more thing that I have to do, like I have to use board. Where's whiteboard, by the way? Here it is. Fine. I can't mute it. Can you see this? My board is visible?
Yes.
Okay, great. So just then after I have to pick up new nucleic acid A level. Hmm. s- Structured questions. Okay. So today I'll pick some hard questions for you. Like we have discussed many of the questions from easy portion and from the medium one. Like today, I think I should take some questions by which you can solve that. This is nucleic acid, but let me check, is this for A level or yeah. Hmm. Calculate the percentage. This is what we have already done. Describe the two structural forms of DNA nitrogenous bases. Interpret the sequence of amino acids from base position 109 to 114 using the key in figure 1.2 below.
Have you pasted the question?
Muscles and stalls. Yeah, I'm going to paste for you. Don't worry. Just give a moment. The question is that I'm checking for you is from so this need. Okay. So Abdul, here's a question. Like you just read out this first and then I'm going to discuss about it. I'm pasting here. Yeah, here it is. Look at this question. And remaining part of it, I'm also going to add up here. It's for 2 mark question, 2 mark question, and 5 mark question. So you would have these specific questions related to this information, which is given to you already on the board, like this one. Look at this. And just please read out loudly so that I can understand
Okay.
where you will be need some help from my side.
Okay. Actually, yeah. So right now I'm reading the big paragraph of the question so that
Hmm.
I can understand what is going on.
Okay.
So some bacteria were grown in the 15N nitrogen culture and then transferred to the normal of 14N.
Hmm.
Okay. After letting the bacteria grow for several generations, the scientists extracted the centrifuged the DNA. During the centrifugation, the heavier, denser molecules settled towards the bottom of the tube. Yeah. Okay. So so the so they are asking where the band is going to appear on on tube B.
Hmm.
So the band is going to appear somewhere around here
Hmm.
because it's not too denser or not too light. So it's in the medium. It should be in the medium.
All right.
So I have drawn and now I'm writing the
Okay. Okay.
statement.
Yeah, you can write the statement, and then after I'll see like, is this correct or not?
Yeah. क्या हुआ है? Yeah, done.
Hmm. What's the key here? Bacteria grown in a nitrogen 15 culture. Bacteria first grown in 15 nitrogen culture and then transferred to the normal N14 culture. So bacteria grown in N14 culture. All right, so this is your key third. And based on this, what you're getting? Oh, okay. All right, here's the question. Some bacteria were first grown in N15 and culture and then transferred to the normal culture. They were left there with enough time to replicate once before the DNA was extracted. Suggest where the band would appear on this tube in comparison to tubes A and C. This would appear in the middle of the tube such that it will be higher than A and lower than B. Uh, okay.
Lower than C. Sorry. Lower than
The
C. I'm.
mm, yes, definitely that should be between the bands in tubes A and C, at an intermediate position, right? Uh,
Yes.
because that should be intermediate in the sense like, but what's the logic behind it? How you would justify it, like in what sense?
Yeah, this is what we have to write in the part B of this question.
Yes,
So
because there you have
now
to
I'm.
justify your answer to the part that is in the part
Yeah.
A. So how you would justify in the second in part B?
Yeah.
Hmm.
Yeah.
The DNA. Justify your answer. Hmm. Explain how masses and stars experiment demonstrated semi-conservative relation. Replication of DNA concerning the results shown in figure 2.1. Okay, all right. So you can write the answer. Then after I'll check.
Yes.
Hmm. This is done. I mean, it's completed now.
Yes.
It's a 2 mark question, so you need to justify in a way like you need to add 2 points. So tube B contains bacteria that were first grown in N15 medium, right?
Yeah.
And then you allowed to replicate once in N14 medium. You have just shifted the bacteria from N15 to N14.
Yeah.
On the other hand, there's a semi-conservative replication because
Yeah.
we know about already like how DNA is going to be replicated. So which model that is going to follow? So this is also in our mind. So semi-conservative replication means what? Each new
As one
DNA
strand contain the as
old
one strand contain 14 N
that would
and
be
other contain
no
15 N.
the old one will contain what? Whether 14 or
For
15.
14.
Old one because you have shifted from 15 to 14.
Yeah, so the old one will contain 15.
Yes, because the previous environment was the culturing environment was and 15. So it means like half of the standard, I mean, the strand of that DNA that would contain and 15, and one new strand that would be light and 14, right? So therefore, the resulting DNA would be that would have intermediate density, definitely like
Yeah.
the DNA that was grown in the culture of N15 and the DNA that was grown in the culture of N14. So once you shifted from A to B or from one tube to the second, so definitely the resulting DNA has intermediate density. So the band appear between the heavier band in tube A and the lighter band in tube C. Getting? So this
Yeah.
is how you have to answer. And please just relate this with the semi-conservative replication as well. Just to say like you have intermediate density based DNA, it's not sufficient until you're not relating this with a semi-conservative replication, because in order to justify this, it's important to have an idea about the semi-conservative replication, right?
Yeah, done.
Getting, Abdul?
Yeah,
Okay, then after you have the section C, we need to explain how Mazelson's and Stahl's experiment demonstrated the semi-conservative replication of DNA, although we are talking about here as well, but we can relate this with the previous in order to justify it. So, semi-conservative replication of DNA concerning the results that's shown in your figure 2.1, which is already there. So, it's for 5 mark questions, so definitely you need some detail here as well. So, let's start to write the answer of this question.
yeah,
If you want any assistance, you can discuss. Otherwise, you should write the answer. Then after we will discuss together. Hmm. The bacteria grown in the 15 and conditions were then transferred to 14 and conditions and allowed to replicate. Semi-conservative replicate. It means like you need to discuss the experimental setting by Mazelsons and
yeah,
Stalls, right?
yeah.
Hmm. 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 The extracted DNA strand showed the entry.
Done.
Okay. So now I'm going to, yeah, it's ultracentrifugation. It's fine. Okay. So I'm just giving you an overview on this. And if you think like, if you need some point to add in your answer, so you can. So as we know, I'm just giving you an overview on this. So Mazelsons and Stahl, as you know, they grew bacteria in N15 medium, like which labeled the DNA with heavy nitrogen already. So this produced a single heavy band. That's tube A, which is already there, right? You have already talked about this. So when these bacteria were transferred to N14 medium and allowed to replicate once, just once. So the DNA extracted in tube B that showed a single band at an intermediate position, not at the same level as either uh, in pure and 15 or
Yeah.
pure and 14, right?
Yeah.
So this ruled out the conservative replication. Okay, so
Yeah.
which would have produced two distinct bands, like as we have studied, one heavy and one light. This is
Yeah.
one observation. Further replication in the M14 medium would produce both intermediate and light bands.
Yeah.
What it confirmed? It it is confirming the semi-conservative model where each new DNA molecule contains one original strand and one new strand. Getting? So this
Yeah.
matches the result in tube C. All right.
Yeah. Yes.
Where each new DNA molecule contains one original strand and one new strand. Like so, I mean, uh, this is how we can say like it is justifying this Matheson's install experiment that's demonstrated the semi-conservative replication of the DNA. Got it? That's good. So if
Yeah.
you want to improve anything from here, so you can. So the bacteria grown in the N15 conditions were then transferred to the N14 condition and allowed to replicate. The new generation of bacteria were examined through the extraction of their DNA through the ultracentrifugation. You should write the name of the technique here rather than to talk about the centrifugal force. Is this not fine to say through ultracentrifugation? It's a technique in which definitely we are using the ultracentrifugal, we are using the centrifugal force. So actual name of this technique is ultracentrifugation, and the extracted DNA strand showed the intermediate density of DNA. You can also add one thing more here, Abdul, to say like which is justifying or somehow it is I would say uh, proving the semi-conservative replication. Somewhere you need to add the semi-conservative replication here as well. I mean, intermediate density of DNA means what? It means like the model of semi-conservative replication is justified or demonstrated from the Matheson's and Stolix. But you should wind up this discussion here like show the intermediate density of DNA, which actually prove the semi-conservative model of Matheson's from the Matheson's Stall experiment. Or you can say like semi-conservative model is demonstrated through the Matheson's and Stall experiment. So actually what we are trying to do here, the actual purpose of this Matheson's and Stall experiment was to justify the model of replication in DNA. So definitely just by having this experiment, we are actually proving this. Like the DNA replication is semi-conservative sort of replication. Are you getting this point? So then
Yes.
after show the hydrolysis of this and this. Okay, I'm just pasting one more worksheet for you. If you think the questions are 2, 2, and 4 of this. Water, this, the amount energy released from the hydrolysis 1 mole of ATP is living cells at 57 kilojoule. Explain why ATP is described as a universal energy currency. Calculate the amount of energy. Yeah. So let's try these questions and then we'll see like we are able to do it or not. These are questions for you. Look at this. Visible? Here's a figure 3.1, which is showing you the hydrolysis reaction of ATP to release energy, right? So the amount of energy released from the hydrolysis of 1 mole of ATP in a living cell is 57 kilojoule. Explain why ATP is described as the universal energy currency. And calculate the amount of free energy in kilojoule per mole released from the hydrolysis of zero point 0.7 moles of ATP in living cell. So give your answer to one decimal place. Yeah, ATP is used as intermediate source of energy by all living organisms in this universe. It's for 2 mark question, Abdul. Hmm. You can give some examples as well, like all the biological processes such as muscle contraction, active transport, biosynthesis. So all of these processes are, you would say, assisted by ATP, right? So it provides energy in small, manageable amount and is quickly regenerated. That's also the very important character of it. So it is actually somehow readily available for intermediate use. Got it?
Like I'm finding an example which is suitable for both plants and animals, such as like the transport, active transport.
Yes. I'm saying like you can add these three: muscle contraction, active transport, biosynthesis, like the photosynthesis process. So all of these different processes, they required some amount of energy in the form of ATP, right? Yeah, that's fine. Hmm. All the biological reactions such as active transport, biosynthesis require energy in the form of ATP. That's fine. Then after you can go for to calculate the amount of free energy in kilojoule per mole released from the hydrolysis of zero point 0.7 moles of ATP in a
You know,
living cell.
I think shouldn't we write biological activities because active transport is not a reaction.
But it's a biological process. We are
Yeah,
just
activity or process, same thing. Like, yeah,
yes. Any chemical reaction, definitely it depends whether that is exothermic or endothermic. So there we need energy, but I'm talking about the and as any process that's going on in the body of an organism, right? So we can generalize this as well. So then after you have the second question, it's of also a 2 mark question. So calculate the amount of free energy in kilojoule per mole released from the hydrolysis of zero point 0.7 moles of ATP in a living cell. So give your answer to one decimal place. So energy released from 1 mole of ATP is equal to what?
57 kilojoules.
And what you need to do here is like if you want to go for
Multiply
energy from zero point 0.7 moles, so then definitely you have to multiply it with that. And
57 times 0.7.
yes, at the end of that, you will get the answer. And what's that? The final answer?
39.9.
Yes, 39.9 kilo, but don't forget to write the unit. That's important. Kilojoule per mole, right?
Only kilojoules, not per
Kilo
mole.
per mole because you have multiplied with zero point 0.7. That's mole. Zero point 0.7 moles.
No, the question is asking the amount of free energy. Oh yeah, okay, got it.
Are you getting in kilojoule per mole? That's already in the question. Calculate the amount of free energy in kilojoule per mole, right?
Yeah.
Okay. Then after you have the next, that is compare and contrast the structures and functions of ATP with ADP. And it's a 4 mark question. It means like you have to give 2 points on structure and as per according to the mark scheme, and 2 points on the function.
According to the equation, like the sign is kilojoules per mole. So it shows that we will divide the energy by mole, isn't it?
By
But we are by
no
mole
no
like
no,
50.
it's a multiplication because the question is like you have to calculate the amount of free energy in kilojoule per mole released from the hydrolysis of zero point 0.7 moles. You can't divide this zero point
Yeah.
0.7 moles because we are trying to find out the energy from this. So whatever the
Yeah.
total amount of energy is there, so you have to multiply it with zero point 0.7. So whatever the value that you will get, so that would be the free energy in kilojoule per mole. So we can't divide this with,
Yeah.
but here we can just multiply, okay?
So next is saying compare and contrast the structure and function of ATP and ADP.
Yes. And it's of 4 mark question.
Yeah.
So you need to add 2 points
ATP
for
has
contrast and
three.
like 2 points for structure and 2 points for function of ATP and ADP. ADP has 3 molecules of phosphate, whereas ADP has 2 molecules of phosphate. Okay. When you are talking about compare and contrast, so we need to talk about the similarity as well,
Differences.
difference and
Yeah,
similarity
yeah,
both,
yeah,
right?
yeah. I have written that both have one adenine base and one ribose sugar.
Great, right? Yeah,
Is it ribose or triose? Ribose, yeah.
ribose,
Oh,
ribose
yeah,
sugar, ATP or ADP. It's a nucleotide.
yeah.
Hmm.
And now we should talk about the
Function.
functions.
Yeah.
So ATP loses
You can talk about its energy first. Like we can say the ATP has more energy, stored energy as compared to
store.
ADP, right? Because then after we can talk about like ADP can be converted back to ATP, as you know, during this process. Like if you have phosphate group there, so then by the addition of phosphate group, you during the process of respiration, we convert ADP into back to into ATP, right? Getting Abdul?
Yeah.
Great.
During respiration, yeah.
Yes, during the process of respiration. Yes, during respiration.
Aerobic respiration.
Yes, definitely.
Yes.
Done.
Okay. Yeah.
Yes. So at least two ADP can be converted to ADP by addition of, yes, 2 points. Definitely two for structure and two for function. So this is how you can get the 4 marks overall, right? Then after we can
Yeah.
go for to have questions like the DNA sequence coding for specific protein was analyzed. The base composition of the template strand is given at the table. Deduce the number of bases in the complementary messenger RNA strand. Calculate the percentage. Yeah, just a moment. It's for how many marks? 5 mark question. Yeah. So, so another question that we are going to talk about. Look at this. This one. And the last question based on this for DNA sequence coding. So that's I'm going to add with it. Explain how transferrin is used in the translation stage of protein synthesis. Hmm, okay.
I think it should be all same. Just we will replace thymine with uracil. That's all the numbers are gonna be same.
Hmm, okay. Hmm. Because messenger RNAs complement with the DNA template strand. Like in DNA, if you have A, so in messenger RNA you would have U. In DNA you have thymine, so in messenger RNA you would have adenine, right? So DNA, if cytosine, then guanine. This is how you can go with it. But the point is, you have to find out.
Yeah.
So from the DNA template strand, you must know like how many adenine. That's 25. Messenger RNA, you have uracil 25. So how you can answer this?
Yeah, I have
20,
answered it.
25,
Oh,
20, 19, 46, 29. Yeah, that's fine. Hmm. Yeah, exactly.
now they're saying to calculate the percentage base composition of
Hmm.
thymine in the DNA.
Um,
Okay,
Deduce the number of bases.
base composition.
Hmm.
So, saying they are asking about the thymine with I mean the bonds, yeah.
Calculate the percentage base composition of thymine in the DNA. Give your answer to one decimal point. Calculation, definitely you need a total DNA basis, right?
Yeah,
First,
yeah.
you have to find out that.
No,
And
no,
once,
like it is saying base composition. So,
no,
does it mean that?
no, let me tell you about. No, no, it's a percentage of thymine. It's a percentage of thymine that you have to find out here. But for that purpose,
Yeah,
first you should know the total DNA basis there, right? So then
yeah, I have.
after,
Yeah,
and then
it
after
is
you,
119.
and the percentage of thymine, then you have to find out the total number of thymine there. And that's very simple, like obtain our total divided by, sorry, multiply 100. So whatever the answer that you will be, that would be actually the percentage base composition of thymine.
It's 24.4%.
Why you're not writing here?
Yeah, I'm writing. I was on my calculator. Now
I'm
I'm
in class,
writing.
sir. Hmm. Mm-hmm. Mm-hmm. But write the answer. I mean, is this like give your answer to one decimal point? Okay, that's fine. But
Yeah,
we don't need any to show our working here.
yeah. I'm showing. I'm showing.
Yeah, that's why I'm saying like you should also show that you're working as well in the question because it's a 2 mark question.
Done.
Okay.
mRNA provides a template for protein synthesis. Protein synthesis requires tRNA molecule. How the tRNA is used in the translation stage of protein synthesis. So it
It's a
it brings
it,
the
it's a 5 mark question, Abdul. So means
oh
like
it's
you
a five
need
mark.
detailed answer. Yeah.
Okay.
So what you need to talk about, like all that
Translation
process of
process.
translation process, how that transfer RNA is used in the translation stage of protein synthesis. First, you can talk about like each
When
transfer RNA
but
molecule carries a specific amino acid. Yeah. Yes, Abdul.
yeah, I'm writing.
Okay. When the messenger molecule joins ribosomes, the transfer brings a specific amino acid according to the template triplet covalent code of the mobile stores. Hmm. 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 Done?
No.
Okay. Hmm. Great, great. Hmm. Okay. But don't forget, like, explain how transfer RNA is used in the translation stage of protein synthesis. Our main focus in every
Yeah,
point should be the transfer RNA, right?
yeah, I mentioned transfer RNA, that how it binds the, how it
Hmm.
leaves the ribosome then.
Okay. All right.
Yeah, I'm done.
Okay, the process stops when stop codon is translated by transfer RNA. Okay, fine. So here, definitely, each transfer RNA molecule carries a specific amino acid because a specific transfer RNA is specific for amino acid. So transfer RNA has an anticodon region that is complementary to the codon on the messenger RNA strand. Yes, that's fine. You've added. So during translation, the transfer RNA binds to the codon via base pairing in the ribosome. This is what you can consider here. So the ribosome joins the amino acids together to form a polypeptide chain. Yeah, that's fine. Let me check it here on this side. Hmm. So each time two transfer RNA molecules are present in ribosome, which joins the amino acids when one empty transfer RNA molecule leaves ribosome. Next molecular transfer RNA comes inside ribosome. This process is called elongation. This process stops when the stop codon is translated by transfer RNA. Yeah, that's fine. I think it's for 5 mark question, so we have 5 points at least here. Hmm. Brings the specific amino acid according to the triplet. Okay, that's fine. It's good, Abdul. So this is how we can answer these questions. So the one last question that we can talk about, and most of, I mean, the questions from here, the nucleic acid that we have covered. So one question that I'm going to put for you is for 6 mark question. That's a transcription is the first step of protein synthesis in cells. Discuss the similarities and differences of the processes of transcription and DNA replication. That's very important to have the differences, similarities and differences. It's a 5 mark, sorry, 6 mark question for you. And straight the first transfer, yeah, that's fine. So this is the last question from this section. Here it is. Look at this, Abdul. Transcription is the first.
There was another important thing I wanted to talk about. Like, I was studying in my textbook, and there was written that, during transcription, when the strands of the DNA are broken, they are not broken by RNA polymerase. They are only joined by RNA polymerase. They are broken by DNA helicase actually in transcription as well.
Are you talking about the transcription?
Yeah, when the strands are broken, they are broken by DNA helicase, not RNA polymerase. RNA
RNA
polymerase.
RNA
They are,
polymerase, definitely helicase means function is to open the chain, right?
yeah, yeah.
In case of replication, this is quite prominent, but how like it is also in case of transcription, right?
Yeah, yeah, it was
Transcription or
written in my textbook.
replication. That's
Transcription and DNA replication both. DNA helicase is used.
okay. That's different. But you have to find out whether in both in prokaryote and eukaryote, how that is differing here, because we need to know the difference of both, like the prokaryotes and eukaryotes as well.
But in my textbook, it's written that the enzyme DNA helicase break the hydrogen bonds between the bases in a specific region of the DNA molecule.
Hmm.
It's written below the transcription in my
Hmm.
textbook.
But how you can differentiate whether it's for prokaryotes or eukaryotes?
I think it's for both. For both, it is same for both.
Hmm. In both cases.
Yeah, in both cases.
Mm-hmm. Let me check it. The enzyme responsible for opening the strand of DNA during transcription. Can I check it? I mean, how can you just send me the screenshot of that? Is it possible for you to send me that specific portion of it, like how it's mentioning that transcription is responsible for opening the
Yeah,
or unwinding this strand of DNA? Just send me the screenshot here on the board. Like, is it possible? Or show me.
helicase is responsible. Yeah, sir.
Hmm? Helicase is responsible in case of replication. That's very prominent. But I'm talking about the transcription process. In transcription process, how helicase is involved. That's the thing that I need to confirm.
Yeah, I am sending you the picture of my textbook, which
You
shows,
can send me
which
my
tells
chat
this.
box as well. But that should be in transcription. Keep in
Yeah,
mind
yeah,
the process, the topic should be of transcription, like all of the steps which are there.
yeah, yeah,
Hmm.
yeah, yeah.
That's in case of replication, but in transcription, RNA polymerase itself carries out the unwinding. That's a confusing statement.
Okay, actually, I'm loading my WhatsApp because I will send. Uh, just give me first. I will write this question, then
Okay,
or I
okay.
can
It's fine.
or I
We
can
have
just.
we have short time here because we have to wind up this as well. But keep in mind, this is important thing that you need to confirm from your side as well. Somewhere that if you are using some notes from there, so it's possible like that that's a mistake there, okay? But if you have the standard book, you can just send me its reference as well so that I can check how helicase is involved in case of transcription. Yeah, it's quite clear in case of replication, the helicase enzyme is involved. But you should complete this answer first. Discuss the similarities and differences of the processes of transcription and DNA replication.
So describe the similarities and differences of the process of transcription and
Yes, it means like it's of 6 mark question, so at least you need to add uh 4 points for similarities and
yeah.
about 4 points for differences.
The one difference is that DNA is unwinding in both processes.
Yes, unwinding is common in both of cases. In both of these cases, yes.
Secondly,
Hmm. Hmm.
secondly, the complementary
Complementarity, yeah, that's also one of the keys.
strands are made during both
They're complementary
processes.
based pairing, actually. You would say like both use DNA strand as a template to build a new strand using complementary base pairing, right? And what do you think about enzymes?
Yeah, it's like ambiguity here right now, so I'm not
No, no,
mentioning
no, no,
that point.
no, but the one thing is not ambiguous. That is, both processes are catalyzed by enzymes. RNA polymerase in
Yeah.
transcription, DNA polymerase in replication. That's quite clear. The confusion is only in case of, like, as you said, DNA strands are opened by one enzyme that is helicase in both of these cases. Definitely helicase enzyme, its main function is to open these strands. But I just want to
I'm
check like
writing a
whether the helicase is also involved in transcription. However, it's quite prominent in case of replication. So yes,
I'm
what do you
writing that in
Mhm.
transcription the DNA sorry in the RNA polymerase is
Mhm. RNA polymerase in transcription and DNA polymerase in replication.
yeah, that's what I'm
So
writing.
the point is that both processes are catalyzed by enzyme. This is one thing we should keep in mind. And one more similarity, I think that can also be added here. Like both of these processes are taking place in the nucleus of eukaryotic cells. That's another similarity that we can take if we are talking about the similarities.
So you're right that the RNA polymerase joins the nucleotides,
Hmm. Yes.
forming phosphodiester bonds in RNA.
Yes, that's another similarity that you can add. And let's get back to the differences now. Oh, yeah.
This is a different difference because in it's DNA polymerase in DNA replication.
Hmm. And double standards, single standard. That's also transcription produces a single stranded messenger RNA, as you know.
Yeah.
While replication produces a double stranded DNA. And there's another difference of nitrogenous base. Transcription uses the base uracil because that has to synthesize the messenger RNA and instead of thymine, while replication uses thymine, right?
Yes.
And what's about like another difference is like only one strand of DNA is copied in transcription, like the template strand is only one strand, and in replication both strands are copied in replication process.
Yeah.
And the one more prominent difference is, as you know, transcription is primarily it's important for the protein synthesis, right, Abdul?
Yeah.
And what about the replication? That's for what's the main purpose of replication?
Sometimes my top for the cell division.
Yes, for the cell division. This is what you can add here. So this is how we can differentiate both of these DNA, transcription and replication process, right?
Yeah. After that, I will send you the image of my book.
No, that's fine. Which book that you're using? Can you just refer me the author's name as well?
It's Marian Eisen. It's the official book from the WJAC Education. It's a official
Yeah,
book.
you can send me. You can send me its cover page as well. Okay,
Okay. Just 1 minute, I will send.
it's okay. It's okay. You are sending me on like in the Zoom
On the
chat.
Zoom channel. On
Okay,
Zoom chat, yeah.
okay, fine. So meanwhile, I'm going to conclude here, and you can ask to Abdullah as well to join the class. I'm
Yeah, he's joining
just
in. Yeah.
in. Okay. Okay, Abdul, goodbye from right now. So I'm going to close your recording from here, and
So.
you can send me meanwhile. So